Hello, everyone, and welcome to today's webinar, Antibody Titration for Flow Cytometric Analysis of Extracellular Vesicles. Are cell-based protocols up to the task? I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Milton Biotech. Now, we encourage you to participate today by submitting as many questions as you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and thus offers free continuing education credits. So please click on that continuing education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speaker, Dr. Desmond Pink, the Chief Scientific Officer of Nanostics Inc. Desmond, welcome. You may now begin your presentation, sir.
Thank you. Good day, everyone. Welcome. Today, I just want to give sort of an agnostic approach to antibody titration and how it's used in the analysis of extracellular vesicles or small particles in general using microflow. So just some disclaimers. I'm a founder and employee at Nanostics, not received any compensation from Miltini for this presentation, and Miltini has not directed any of the analysis or censored any data from this presentation. The views are my own and do not necessarily reflect the views of Miltini Biotech. So I just want to introduce the team. This is my research team and my clinical team, Michael and Diana, Juliana and Arga. Also, I'd like to introduce Asia Riegers. She's helped me do some of the imaging flow cytometry here in this presentation. And of course, the big boss, John Lewis. So what I want to do today is sort of give you an overview of some of the things that I've found over the years are important to consider when performing antibody titration. And it should be a fundamental exercise that you do in your lab, especially when you're undertaking any new cohorts that you're going to be analyzing, any new reagents. Basically, it should be something that you do all the time. So we'll touch upon some samples, reagents, tiny bit about instruments. We'll do a lot about the analysis, and I'll also talk about some of the remaining questions that are always sort of popping up. But one thing that I really want to do is really balance the theory with some practical real-world experience so that I can put my money where my mouth is. So starting off, so why even bother titrating antibodies? And I ask this question because I think we're all taught, titrate your antibodies. But I've had so many collaborators and so many students that I've worked with who just simply have never either heard about it or don't know how to do it properly, or even appreciate why you should do it. So the classic answer is to basically get the best fluorescent measurement. You want to maximize the signal while reducing the noise. So in a prior life, I did a lot of microscopy. So what I'm showing you here is sort of my visual interpretation of what titration would look like for a microscopist. So this is an image of a highly vascularized tumor with an antibody not titrated. Titrating it without the absence of your treatment or your drug, we can actually see a lot more clarity here now. We can actually see the vascular system permeating into the tumor. Now, actually, optimizing your antibody titration allows you to see very small changes or rare events, if you're talking about flow cytometry, when you have your antibodies properly titrated. So again, not titrated, a lot of signal in the background. Titrate your drug, you actually minimize that noise, and then it allows you to see very small changes. So again, one of the other parts of why even bother is your data reproducibility. I mean, blaming it on antibodies and the discord in being able to validate whole studies has been blamed on antibody irreproducibility for a number of years. This is 3 Nature articles spanning the last 5 or 6 years, and this topic comes up all the time. And one of the things that really comes out is quality. And it's not sexy. Doing an antibody titration is not the pinnacle of research, but it is crucial. So whose responsibility is it? Vendors need to be able to provide you the antibiotic product that they state in the vial, but the onus is really on the researcher to make sure that that antibody performs as you expect it to perform. So what are companies doing? So because Miltenyi is sponsoring this, I'm going to plug them for this. And Miltenyi, like other companies, they have introduced programs over the last number of years where they're doing recombinant antibody production. And what does that allow us to do? It allows us to decrease interfering binding on the Fc regions, allows us to improve lot-to-lot reproducibility. The purity is better, and it allows us a bit more flexibility if we're going to engineer new components. So all of these things really help consistency over the course of your project, especially if you're a master's student or a Ph.D. student or a trainee, and you're doing studies that span years. So next, the question is, when should you titrate? And I think everybody wants to say, the first time you buy an antibody, you should always titrate it. And that's true. First time you use a particular clone or conjugate, obviously you need to titrate this. We've also found that if you have prolonged storage, and especially if you're in a lab and you don't know who else has been using it, if you have a summer student that comes in and potentially contaminates the stock, just check it. Run a simple titration. It'll save a lot of headaches. If you get new or suspicious shipments of your antibody, so by suspicious shipments, I basically mean, I live in Edmonton, Alberta, Canada, and at times it can be 35 degrees Celsius in the summertime and minus 35 degrees Celsius in the wintertime. So there are chances that our antibodies could freeze. So if you get a weird looking shipment, titrate your antibody. It'll tell you if it's working properly. If you get shipments with different lot numbers, titrate. If you have a long time between orders, even if it's the same company, same clone, and same conjugate, you got to realize that antibody companies are going to make big batches of these antibodies, and they're going to be sitting in vats. Purified proteins will degrade over time, so take that extra, couple of hours and titrate to make sure that your antibody is behaving the same way. If you're getting it from a different company, if it's the same clone, same conjugate, you don't know what's happened if that antibody is coming from a distributor or from a supplier to a different distributor. You don't know if there's been weird shipment anomalies. So just take take that half morning and do it. And again, if you get suspicious data, so we always try to have positive controls for our antibodies. And if your positive controls fail, and even after you've done the titration, like weeks beforehand, go back and just do a quick one, and it'll tell you a lot of information. So we've experienced pretty much all of these. And while I'm not going to show data for all of these, if you guys want to see data, just give me a shout and I can provide data for this. So what does a titration analysis look like? If you Google antibody titration, these are pretty much the typical graphs that are going to come up or pictures that are going to come up. And these are fine. This reflects what you're going to see in cells for the most part. Is this sufficient for EVs? So when we talk about actually, looking at noise, so the noise would be down on the bottom of the left-hand panel, and then you can see your antibody fluorescence going up. What does this actually look like? This is essentially the stain index. Your separation between noise and antibody signal is what you're measuring. And you basically want to look at the spread of that data. So the high peak here on the right-hand side is basically your antibody signal on your positive events. And the small part on the bottom is basically the background or the noise. So this gives you your stain index. So some people will use stain index. Other people will use separation index. And they're very similar. And there's a whole series of papers that describe the evolution of these equations. And I would strongly encourage you to use these equations. They can really help you make decisions about your optimal antibody concentration. So that's what it looks like. That's kind of the math. What does it look like when you graph it? So what I did is I went and I Googled a lot of the titrations and looked at a lot of the presentations. And essentially we came up with, there's three classic scenarios. You'll get your stain or your separation index that kind of look like Michaelis-Menten kinetics, if anyone can remember their enzyme kinetics, or your variable dose response here in the middle. And there are times too when you can get substrate inhibition where at higher concentrations of your antibody, it looks like the signal starts to come back down closer to the noise, or the noise goes up. And so the separation between those two peaks decreases. So let's talk about some real data to look at the protocols, some do's and don'ts, and how we actually get some real data into this. So we've kind of worked out a sort of a general scheme that we use to test all new antibodies that come in, or antibodies that we have going for different cohorts that we use. The first thing we do is actually screen for positive samples. So we use a lot of primarily plasma and serum-based human samples. So there's a lot of variability. So it winds up being, for some antibodies, you get all or none responses, and some more you get graded responses. But you always want to have a sample that you can use in order to get an adequate titration. And this will become more clear in the next slide. Your assay design. This is something I really strongly encourage everyone to do, especially if you're starting out at your the beginning of your program. Make sure that your titration range is adequate. 3 concentrations is not enough. You basically want to span a good range, maybe 100-fold for your antibody. We always use the manufacturer's suggested concentration or reaction volume as 1x, and we go from there. Typically, we do 15 to 30 minutes incubation time, and because we're designing most of our work for clinics, we always assay at room temperature. Room temperature is designated as 15 to 30 degrees Celsius. Standardize your diluent and your dilution depending on the platform you're using. But most importantly, standardize this protocol for all of your antibody titrations. If you do that, if you can standardize it, then if you have a weird problem that shows up, you can backtrack a lot easier and kind of figure out where things may or may not have gone wrong. One thing that we've actually found extremely beneficial is we actually have a standard form that we use for titrations, and this works really, really well. In terms of screening, I'll make a note here. So we've used a fasted sample to analyze CD41A. That'll become more apparent in a couple of slides. Execution, if possible, automate it. Okay, lots of times you're going to be dealing with very small volumes of antibody, and antibodies are sticky proteins, and they can stick to the tips of your pipettes. And a little bit of 10%, 20% error can go a long way in the data. We'd like to perform all of our assays in triplicate with at least anywhere from 60 to 120 seconds of assay time on the flow cytometer within the paradigm of we're not in swarm for the events, and everything is clean. You also want to make sure that you're assaying enough positive events to actually get valid data. Okay. For the analysis, I strongly encourage people to plot your stain index and your separation index. Most times they'll look pretty similar, but there are times where they look weirdly different. Plot the MFI, your median fluorescent intensity, of both the positive and the negative populations. And as well, plot your positive event concentration. You want to see all of these things. That'll educate you as to what the best antibody concentration you should use will be. For these, I typically use a mixture of software packages, FlowJo, Histogram, FCS Express. There's probably a bunch of other ones out there. These are the ones that we just typically use. And then finally, evaluate. Use stats and graphing to evaluate your analysis, not just the visual. And the beauty of actually standardizing your assay design, you can pull upon historical data, especially if you're running the same antibody titration over the course of a number of years. We've done this for various groups, and it can make a big difference. If your samples are sitting in the freezer for a year or 2 years, or your antibodies are in the cooler for a couple of years, it makes a big difference. So this is just a screenshot of an example of staining or screening for positives and negatives. So in this one, we typically use, what we have here, 8 to 10 samples that we have for screening purposes. And this is what we would test various biomarkers for. And you can see that for biomarker 1, we get 3 hits. Biomarker 2, I'm not getting really any hits. A little smidgen of population coming up there, but not very much. Biomarker 3 is a winner. We've got strong hits in every single sample, and they're all nicely separated from the noise. And biomarker 4, I've got a single. The nice thing about doing this is that right off the bat, you're going to get positives and negatives. Except for biomarker 3, we're just getting all positives, and we can investigate that later as to, is this an interference, or is this antibody clean? Is it noise? There are also a bunch of controls that you run. Isotype controls will give you a mixed bag of results depending on what you're looking at. You can run them, but there's a lot of literature out there that says that they're not as good, and matching them up with your antibody is challenging sometimes. Internally, we've been developing a broader internal negative control for both plasma and serum. And this works for the majority of samples, but we do get some instances where it absolutely does not. And I'll show you examples of that shortly. So when we execute a titration analysis, what kind of data do we take? So to highlight some of these different effects that I'm going to talk about, I'm basically going to talk about using CD41A as a platelet marker and a platelet-derived particle, or PDP, marker. So since we're using the HIP8 clone for all of these, and I've got conjugates that range from the E450 all the way down to PE and APC. We always take our product number, the RRID number, lot numbers, and when they're expired, and most importantly, the stock concentration of each vial. For this experiment, I've also used cell mass green and some Triton X. Sorry, that data didn't show up here. And plasma. We have all of our plasmas categorized and ID'd, de-identified for privacy. So this experiment is basically to highlight the effects of the same clone and different fluorophores. And this experiment was executed on the same sample, same day, same operator, same machine. And it's important to note that platelet-derived particles are present in both plasma and serum. Just because you've clotted the blood doesn't mean that you don't have platelet-derived particles. So what does this look like? So this is a standard microflow cytometry plot looking at CD41A, and this is a 405 excited fluorophore on the y-axis and large angle scatter on the x-axis. So the blue highlights the total sample, our platelet-derived particles, and also intact platelets on the far right. In the course of doing microflow, it's also important that there are a number of controls that you need to run to standardize your equipment. So here I'm just highlighting some of the light scatter beads that we use. So we've used a bunch of the apogee beads. I've used exometry beads, and I've also made my own internal mixture of NIST standardized beads ranging from 80 nanometers polystyrene all the way up to 200. And this is a way to sort of get an idea as to how small can your machine or your platform actually resolve. Similarly, you can do the same for MESF, and for this talk, I'm not going to show this. I'm just trying to get into the antibody titrations. But it is important to standardize your equipment to make sure that your equipment is actually behaving the same day to day. You need to calibrate and monitor your machine on a pretty regular basis. So again, to adhere to the MISAV guidelines, you want to make sure that you're actually looking at vesicles. So this is, again, CD41A on the x-axis and cell mass green on the y-axis. On the far left, we have basically just the sample. The categories here are just below the histograms or the cytograms. Just the sample. The next one, we've got the sample plus the antibody, and you can see that we're starting to get the CD41A positive platelets and platelet-derived particles showing up. There's a little bit of green fluorescence because the big particles will actually bleed into the green channel. Adding in the cell mass green, you get a significant shift. Now, it's important to note here that depending on what you use, you should titrate your cell mass green for incubation time, final concentration, and temperature. All of these things can make a significant impact. Also, depending on the antibody, you want to make sure that the addition of cell mass green doesn't interfere with antibody binding. So we've done that in separate experiments that I haven't shown here, but we've noted that there can be interferences. And the same, adding in Triton X100 in the far right cytogram, you can see that the data looks completely shifted. And again, depending on your sample, you may have to increase the incubation time and temperature of your Triton X100 because you may have significantly more particles than you're used to seeing, especially if you go from cell culture to plasma samples or serum samples. The concentration of particles, so you just may need to use more detergent. So we've got the controls all done. This is just a graph to show you, here's all the data. This is what you would look and see on the screen, ranging from the 405 excited all the way down to 638 excited. So when you plot it, what do you see? So essentially, you know, if I replicate the images I would get from Google, this is what I see. But for the total, you know, the negative population in microflow cytometry is going to be predominantly negative, or sorry, predominantly the bigger population by 90 to 95, 98%. So the populations you're interested in will always look very, very small. This would be your platelets showing up just as little tiny hills versus the mountains. The platelet particles you actually don't see on this scheme. And platelets, if I gate just on those, you can see them a bit more clearly, and you can see where I've made the cut. But again, it's really hard to sort of make a decision. So is this really helpful as opposed to actually plotting the data? So this right now is our standard, whenever we do a titration, these are the standard 3 graphs that I look at. On the far left, first thing I do is plot MFI. So on the bottom in the clear circles, I get the MFI of the negative population. And you can see that it does raise or increase above the starting concentration a little bit, but we're going from maybe an MFI of 200 up to about 300, so not too much. Whereas the positive population definitely significantly increases and plateaus. In the middle, we're looking at the separation and stain index, and both of these in this scenario actually agree. And what I do when I plot these, and I haven't identified this, but I use a program called GraphPad Prism to plot all of my data. So it allows you to do nonlinear regression fairly easy, and you can compare different regression analyses. So I typically will always compare substrate inhibition to the variable slope, and that takes care of, I would say, 95% of the analyses. But you can see here that the index peaks and then goes down a tiny bit. One thing to note here is that this is for the platelet-derived particles, and the separation index is only at around 7, as compared to most of the cell-based protocols where you're looking at 20s and 50s and 100s. And again, I also look at the concentration here. I've shown you both counts and concentration, and these similarly plateau. This also tells you, when you plot the concentration, that you're actually seeing enough events to actually make an educated decision about your optimal concentration. So moving next, this would be the actual data for these 5 fluorophores. So I won't spend too much time on this other than to say that each of them look fairly similar. There are differences between the platelets, your total, and your PDPs. You'll see that the plateaus look pretty similar for the separation index. but sometimes the graphs may be a little bit different. You will see that for this particular one that I've enlarged, I can see a population in each of the cytograms that shows up in the upper right-hand corner. And for this particular analysis, this was some aggregation that we saw with the actual antibody at high concentrations. So it's also a way, don't just plot the data, look at the actual cytograms as well. So I have to thank Asia for this graph, because after I did the analysis and plotted the optimal antibody concentration, they asked me, well, do you get the same numbers for your platelets and your PDPs? Platelets are basically what I'm using as a surrogate for cells. And I didn't see any difference in these particles. Basically, these all raised up as soon as the antibody titration got high enough. They all gave me the same data. The PDPs, however, actually did significantly increase. And if you plot them relative to the conjugate brightness, and basically what I did for the E450 and super bright, I put in the sort of the relative brightness of where I expected them to be. But when you plot them this way, it actually makes sense that as you increase the brightness of your fluorophore, you may see more particles, because more particles are actually getting above that threshold of detection. Now, this is where your MESF may come in, and I'll standardize this. But the take-home message for this slide is basically, your large particle detection is going to be less affected by your fluorophore brightness than smaller particles coming from that same cell. And again, this is sort of one of those graphs that you have to do. I mean, if you titrate your antibody, you can save money. In each of these particular antibodies, we're actually using significantly less than the vendor recommended concentration. So it does pay to actually literally pay to titrate your antibody. Now, there will be some situations where you actually have to use more the concentration. So let's move now a little bit about graphing and presenting your data. So I use a variety of platforms. I've just highlighted them here, FlowJo, and the text that I put here, there's some very good references. FlowJo Exchange basically talks about using a R plugin to automate the separation or stain index calculation. It works best if your EVs are very well separated from the noise. There's a couple of YouTube videos for FCS Express, but I will strongly suggest you use the resources from the University of Wisconsin-Madison. That has been an excellent tool, especially if you're new to using FlowJo and you just want some help and guidance moving through that. So what does it look like when you actually graph? Oh, before I get to that, one thing that I do want people to look at is if you have a situation where your EVs are starting to separate from the noise, but you can't quite tell if they are, the resolution that you see in some of your software packages may be hindering that. So I know in FlowJo, it's typically a 512 by 512 sort of scenario. In FCS Express and histogram, I can go from 512 to 512, but increase that up to 1024 by 1024. And that can actually make a difference, especially if your population is very close to the baseline. It's just something to keep in the back of your mind. So again, how does this start when you're doing your graphing in FlowJo? In panel 1, you're basically bringing your data in, making your tables. For here, this would be the CD41A, so I'm basically making platelets versus, I'm sorry, no, this is not the CD41A data. This is something I stole from work I was doing a couple days ago. But since you bring in your data, panel 2 shows you sort of the gating strategy. Essentially, I gate as much of the positive and negative as I can to be as unbiased as possible. From this gating strategy, you're going to then make a table with different columns, and each column will talk about your MFI. And in number 4, this basically shows you the formulas you would use for stain index. And this is all spelled out in some of the previous references I showed. And then at the end of it, you can actually generate your table that you can put here on the clipboard. One thing to note with the tables that you generate if you pop them into Excel, you can then use that to orient your data in a way that you want to for graphing. But I have noticed that this data, and it just may be the way that I've got my computer set up, will give you commas instead of periods for decimal points, and that can confuse the program sometimes, so you can just go in and correct those. So taking that data and graphing it, some do's and don'ts in my opinion. If it's XY data, concentration, independent, dependent variable, plot it on an XY graph. Don't plot it using bar graphs. And I say that because you can misinterpret or lose some of the information that the data is providing you. For one thing, small ranges in the concentration of your antibody can actually be skewed because it'll take up half the graph when it should only take up, a fraction of that. One thing that I have found with a lot of colleagues and a lot of collaborators is when they talk about their antibody concentrations that they're using as dilutions. I would strongly suggest that in your standardized protocol, you actually use micrograms per reaction, especially if you standardize the volume of your reaction. That allows sort of, no miscommunication. You know how much antibody you need to have per reaction or test. And I also would strongly suggest doing more than 4 concentrations. I like to use a wide range, so about 100x. And when you look at the data, be your own worst critic. Look at the data that you've plotted and go, did I screw something up? And if you did, take that and move with it and fix it. This first pass will also tell you if you need to include more concentrations at a lower range or a higher range. And again, when you plot the data, don't just use the first plot that comes out on your software. Take the time to look at and change the data so that you get a really solid analysis of the data and it speaks to what the data should be showing you. These are 4 graphs showing the exact same data set, and all I've done is I've changed either the X or Y axis from linear to log. That's all. So the data looks very, very different, but you pick the one that tells you the information that you need to actually use to make a decision. So just sort of summarize in this section, why titrate? I mean, you're looking for the best resolution of your signal to noise, and you're trying to save money and have reproducible data. When to titrate? New clones, new conjugates, new lots, new shipments, weird storage, weird data. Typically, I find just 3 kinds of classic data to expect. Titrate, you're going to screen samples, design your experiments, execute the analysis, analyze the data, and then evaluate the data. I've shown you some real examples with CD41, and I know I've gone through this fast, but you guys can go back and look at the data in more detail later. I've highlighted controls for light scatter and fluorescence, and negative and positive controls. Fluorophore brightness can definitely impact your detection of small particles and EVs. There's a bunch of different software packages for analyzing, but more importantly, critically evaluate your data. So I know that CD41 is a bit of a cheat because it's easy and it's going to give you very clean data. So I'm just going to give you a couple of more real examples to highlight. I've de-identified these for IP purposes. So I'll start off using this one. This was cdxx. This is a BioBlue conjugate. It gave us great titration. This is exactly what I want to see every single day. I don't get to have that, but that's what I want to see. This one was great, but it caused issues further down in the multiplex. So we had to move away from this one and went to another 405 excited. So I tried a different company using the super bright 436. Again, this gave me really nice bright data, but it behaved very differently. In this scenario, the background came up dramatically, and the curve really showed a dramatic reduction in concentration, and the index values also showed a similar sort of like substrate inhibition. So in this one, this was weird. What do? So, I mean, I think the first sort of go-to is that you need to turn your gain down or your PMT. So we set up our gain or our PMT at the beginning so that we get the minimum amount of noise. Again, this data, same antibody, same clone, different companies, different conjugates. But you want to set your equipment up at the beginning so that this is not going to be a problem. And you want to, I think in this scenario, go back and actually assay some lower concentrations of the antibody. That's what we did. So another example of easy data, this is an APC. So again, first couple of concentrations, starting with unstained, moving up to higher concentrations of the data, just following the graphs. You can see, a nice increase in MFI and concentration kind of plateaus. And our internal negative control here was nice and clean. The data looked very similar, plateau's nice. This is what I want all the time. This one is a little bit different, not so easy. So similar type of sample, but the data was very diffuse at most of the concentrations. I didn't get a very nice tight group of EVs. It was almost as if the antibody itself was dirty. But the background didn't go up, as you can see by the plot in the MFI graph. The index values didn't perform as a nonlinear regression. It looked more like a linear regression, which is kind of weird. And of course, our internal negative control down here lit up like a Christmas tree, which occasionally happens, just to show that our internal negative control was not universal, or is not universal, which is fine. We know that going in. So this one required more work. So what are some other things to think about? There are a lot of other questions that we want to talk about, and I'm just going to try to highlight some of these. Your sample complexity and panel building. So cells and EVs, both media are very complex. You're looking at different parts of the whole sample. I mean, cells have a lot of granularity, and so you can separate some different types of cells simply by light scatter. EVs don't separate that way. You can maybe separate platelets a tiny bit, but I'm saying that with quotations around them. Most EVs, I would say 99% of EVs don't separate at all by light scatter in that same scenario. There's a lot of panels for cells for a lot of different diseases. They have been made and tested and validated over the years. EV panels for different diseases don't exist yet. We can look at 3 to 4 biomarkers at a time. I would hazard, and that's for physical reasons, and I'll get to these now shortly. But I think the field is moving in that way, where we'll soon, in the next few years, start to have some panels. Matrix effects. So if you're titrating an antibody and you titrate it for plasma, can you use that same optimal antibody concentration for serum or for urine or CSF or any other media? You know what, you got to do the titration and check it. In this particular case, plasma and serum actually behave pretty similarly. And this is with EDTA and SST tubes. Urine is different altogether. I mean, you have a different concentration of EVs. The pH and dilutions are going to be different. You're going to be looking at different concentrations of particles altogether. And I should say that of all the data that I've shown you, none of these are with isolated. These are simply a small aliquot of the plasma or the urine or the serum and antibody added directly. These are not enriched with beads or any other manner. If you do enrich with any beads or stuff like that, the titration is performed exactly the same way. You're just using enriched particles. So think about your particle size. I mean, we've all seen these figures, but it's important to note that, as your particles get smaller, the number of receptors that you have available for interrogation decreases. That's really important to keep in mind. Just like cells, EV analysis, you need to balance your expected antigen density with your fluorophore brightness, right? So you need to look at that as well. Small particles, they scatter less light. So then also you need to think about your triggers. So all of this stuff comes into play. So let's put this into sort of a context. So this is taken from data from the Zeiss campus and backed up with data from paper, which I apologize doesn't seem to have been referenced here. I will follow that up. But this is a standard IgG molecule, and here you can actually see in pretty much approximately the right size fluorescent proteins attached, synthetic dyes on the bottom, or quantum dots. And you can notice that quantum dots change in size as you go up in the excitation or the emission. So this thing that really shows up here is that, the fluorophore conjugate is actually fairly big in relation to the actual IgG molecule. This is PE. It's, again, just as big as the antibody almost. So now let's put this in the context of your EVs and your exosomes or EVs. So this is COVID-19, which is agreed upon as anywhere from 50 to 140 nanometers. I think most people say it's about 100 nanometers, so right mid-range. The spike protein on top, there's about 300, I think, instances of that. That's the dark purple on top of the liposome-looking molecule. The sort of lighter blue color would be your antibodies. So now you have to think about the crowding of, your antibody mixtures and the availability of just simply accessing those epitopes or those antigens. So let's compare that with a cell. So I found this really neat paper in Nature Immunology that basically said, if a human being is the size or scaled to the size of an antibody, then if you take a lymphocyte and spread it out, the total surface area would cover the entirety of Central Park and large parts of the upper west side and east side of Manhattan. There's a lot of room here for antibodies to bind different receptors compared to the crowding we're going to see under standard extracellular vesicle. So let's also look at your fluorophore brightness. So here I've just presented a 3D analysis looking at the brightness of your fluorophore with the excitation and the emission. And I apologize if this is not clear. It would be excitation. Hard to say if one is facing or not, but you can sort of see it going from left to right. You'd have 405 excited, and more to the right, you'd have the 638 excited. But it shows you, and I've just highlighted like brilliant violet 421 PE, they're bright. Compared to FITSI, which is kind of right smack dab in the middle of the diagram, and as well the Psi-5s and the APCs show up there. So this just sort of to put it into reference. Now the yellow box that I put on the bottom, in my experience, and others may disagree, but I found that these particular fluorophores are difficult to see on extracellular vesicles and exosomes, whichever you want to call them, small particles. And again, I want to emphasize, you need to balance your expected antigen density with your fluorophore brightness, because this can have implications for multiplexing later. So what if you're unlucky enough to have just a simple primary antibody with no conjugate and you want to do a secondary? Well, take into consideration that you're actually adding in another antibody now, and all of these different components move around. So your crowding becomes even more complicated on the antibody. More importantly, in the context of titration, a lot more wells, because you have to titrate both your primary antibody concentration and your secondary antibody concentration. And that's what we want. We all want to do more work. So I'm going to try to rush through this in on the next few, but I just want to highlight standard flow versus imaging just as an example. So I repeated the CD41 assay, ran the same controls, and got the same data. You guys can come back and look at this. I'll just skip to the crucial parts. This would be the raw data for the imaging flow. Using the imaging flow and compared that to the microflow, standard microflow on the apogee that we've used, I got the same optimal antibody titration. They matched. The weird thing was, if you look at the separation index on the standard microflow, it was about 300 for platelets. On the imaging flow, it was about 2,000, so extremely elevated. And I couldn't figure out what was going on. And then I realized that because the imaging flow needs to see fluorescence, it was using these particles that were right at zero. So the calculations basically just artificially inflated the separation index, but I was still able to get the same optimal antibody concentration. Event triggering, light scatter versus fluorescence. So I'm just going to quickly look at the data here. You know, on light scatter, I like using light scatter because I like to see the context. Unstained samples, you know, up in the top left-hand panel, you can see small angle, large angle. And then on the bottom beneath that, you can see that there's no positive signal. Add in your antibody, light scatter looks the same, but your positive particles show up. In the blue in panel 3, you can again see the unstained sample, no particles, and hardly anything. It looks like a clear white screen. Now, if you look at your sample plus your fluorescence, you can see a little bit of noise that I've highlighted in the red there, but you do actually start to see, you know, your positive signals. That's fine. This is just what the data looks like. Moving forward, I cheated a little bit on the way that I actually gauged the negative population, but I was able to get the same antibody concentration regardless of trigger, optimal antibody concentration. Concentrations were basically the same, no MFI differences in the positives, and stain index gave similar but with different numbers as expected. But what I want to sort of raise the question, is this always going to be the case? What if you have a situation where you're triggering simply by fluorescence and you have this scenario where your baseline creeps up? For experienced users, this may be obvious, but if you're new to the field and you're doing your antibody titration, the blue data here is what you would get using light scatter trigger. The black would be what you would get with fluorescence triggering. It looks like an appropriate graph. Your event concentration in the blue shows what we've seen before. In the black, this looks acceptable. It looks a little bit weird, but it's acceptable. So things to think about. So again, just to finish up, cells and EVs, multiple levels of complexity. There's a whole bunch of the points to think about in terms of, you know, don't assume your matrix is going to be the same. Think about your antigen density, fluorophore brightness, probe selection, and multiplexing. All these things come together. Your optimal antibody concentration should be independent of your flow platform, but your data analysis needs to be informed. I mean, the optimal concentration should be the optimal concentration. And triggering, I don't care which way you trigger, just as long as you use that triggering in an informed manner. But for fluorescence triggering, what are you going to use as the negative population? So hopefully I've touched on the majority of these. I may have missed some because I realized I was running long. But Julie from Miltini actually said something to me the other day when we were going through this, that antibody titration is an investment. It's an investment in your time, your future time, and literally in the success of your project. I want to really make sure that you balance theory with your empirical evidence, but most importantly, critically evaluate your data. And with that, I will have a little bit of time for questions. Sorry, I know that was rushed, guys.
Desmond, thank you so much for that informative presentation, and we will now start the live Q&A portion of this webinar. And if you have any questions, please ask now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. And it looks like we already have some great questions coming in from our audience. Our first question, Desmond, is there a best antibody conjugate to use? For example, PE and brilliant violet 421 are very, very bright. Should we just use these?
You always want to choose the brightest fluorophore you can, but typically most of us are going to be using multiplexes. So a couple things, that conjugate may not be available for your antibody. So that may be the limiting factor. It could be something very similar, but typically you have to balance your multiplexing with your choice of fluorophore.
Thank you so much. And now, are you really suggesting that we should titrate our antibodies in all of these situations? And there's actually a three-part question here. And is it really worth it, or doesn't it just use up antibody stocks? My boss really wants to see the difference between these treatments.
So, actually, it's up to you if you do or you don't. As someone who's looked at a lot of data and a lot of scenarios and just simply asking the question, have you titrated your antibody? If your experiment didn't work and you didn't titrate your antibody, then I can't believe that there was no difference between positive and negative or control and treatment. Yeah, I really do. I do suggest that you titrate your antibodies whenever there's a question that it may have changed in any different scenario.
Thank you so much. And again, I want to thank our audience for these great questions coming in. Desmond, how do you calculate index if there are no separations between the background and the staining?
That's a tricky one. So it's almost like fluorescence triggering or not. So typically I would run a bunch of different unstained samples, my negative control. If I'm using isotypes, I'll run the isotypes and look at those. And I'll use all of those different situations as the place where I'm going to put my negative population gate. In a lot of EV analysis, you will find that you won't always get a clear separation, and that makes sense. These particles are small, and they're not going to have very many antibodies actually bound, so it may just simply be not enough signal to get above the threshold. They may be there, they just may be beyond your limit of detection.
Thank you so much. Now, aside from availability, do you use a separation index to determine the chosen fluorophore for a given experiment?
Sorry, could you ask that one again?
I said, aside from the availability, do you use a separation index to determine the chosen fluorophore for a given experiment?
I have in the past, yes. There are different times when you can use separation index if you think you're going to have a lot of antigen density. You can say, you know what, this one's going to be really good. Or you can have situations where you have a population of your particles that are bigger for one particular antibody versus a second group that you're looking at within the same sample that are smaller, and you just sort of balance the dimmer fluorophore with the brighter one on the smaller particles. Dimmer fluorophore on the bigger particles, brighter fluorophore on the smaller particles.
Thank you so much. Our next audience member says, Desmond, you mentioned using microgram per reaction, but why not use microgram per microliter?
So that's fine. So I put in microgram per reaction only because our reaction volumes are standardized. If you're using, concentration is definitely the way to go. Because we're looking at different cohorts and sometimes different antibody volumes depending on what we're doing, I like to look at the microgram per reaction, and that allows me to standardize it. But if you go back and back calculate with concentration, it'll balance out. But yes, definitely the concentration is the way to go. Make sure you talk about either the concentration of the antibody you're adding or the final concentration in the reaction volume. That's the part you need to standardize.
Thank you so much. Now, our next question is about matrix effects. Desmond, isn't it the number of cells you analyze and or the number of receptors present on each cell that matter?
So I don't understand the question in relation to the matrix. So yes, you need to have enough positive stain particles. If you're doing a titration, you need to have enough positive stain particles to evaluate your titration. If you choose a sample or a set of samples to titrate your antibody on, and it looks more like a negative sample as opposed to a positive sample, then your titration may be completely skewed. Same way, if you choose a series of samples that have an enormous positive population versus the majority of your samples having moderate to low populations, you may be skewing in the other way. You want to choose your screening samples to be reflective of the majority of samples you're going to look at. And I don't know if I've answered that question. I can follow up if anyone wants further clarification.
Wonderful, thank you so much. Now, what would be your strategy to titrate antibody if you know that your cytining gives you two distinct positive populations? Which population would you use for titration?
So I've tried to highlight that with the platelets and platelet-derived particles in the CD41 data. There are times when it gives you the same optimal antibody concentration. If you have two different populations, the question is, is there something in your, if you're using cell culture, your unconditioned media that's giving you protein aggregates? Is one population protein aggregates? Are they both viable vesicles? There's a lot of questions to answer there. You can actually just do a gating strategy and see if those populations titrate the same or differently. But that's a tough one that you will probably come across.
Thank you so much. And again, thank you for these great questions coming in. What internal controls have you used for the titration, Desmond?
So we have built a lot of different types of internal controls. We screen a lot of patients and a lot of normal healthy controls because we're looking at disease versus healthy. We also have in place a way to actually build our own internal controls. I've used isotypes, a bunch of different isotypes from different companies. Everything that I can think of, I've tried. Your choice of negative control is tough. Building a set for your particular antibodies is not trivial. Not trivial, because it's not only finding a negative population, but it's a negative sample. Finding a negative sample or a positive sample that you can use as positive and negative controls throughout your set of experiments. It's not trivial.
Thank you, Desmond. And we have time for a couple of more questions. Desmond, what do you think is the maximum number of antibodies you can multiplex for EV flow cytometry?
You know what? I've done 3 is my go-to. I've done 4. It is really, you really have to pay attention to the samples you're looking at. Compensation with EVs is not quite standardized for small particle flow right yet, and that does make it extremely challenging. Would I do 5 or 6 or 7? I don't know if there's enough data out there yet to say that we can use the same strategy with EVs as we do with cells where we can negatively gate. I don't know if that data is sound enough yet to actually do that kind of extended panel design.
Thank you so much. And finally, our last question that we have time for today, which is the best Titron 100x concentration recommended? And what about time of incubation?
You know what, that's an empirical one. You got to titrate it. That's a really good question. If you find you've got cell culture, titrate your Titron 100 concentration. Start at a high one of even 5%. You're going to find with different detergents that you're going to have a critical micelle concentration, so you could actually make more particles. Change the temperature, change your incubation time. All of these things influence the amount of solubilization you have. So it's one of these things. I mean, titrating your membrane dyes, titrating your detergents, all of these things inform you about your samples and your actual assays. If your samples are all coming from fasted patients versus fed patients versus cell culture, the stock concentration of particles within those samples, as well as the types of particles within those samples, are all very different. And all of those will affect the way that your detergents work. And again, think of it, if you're trying to solubilize a very cold thing, fat's not going to solubilize in cold very well.
Desmond, thank you very much for your presentation today and for your time. Would you like to provide any closing remarks to our audience before we go?
Just the last thing, I mean, antibody titrations can save your project. It's going to be a question that your reviewers are going to ask. They should ask. Critically evaluate your data. Try to be your own worst critic or your own harshest critic, and that typically will save you lots of pain in the end. But there's a lot of us out there that have done a lot of these types of experiments, so that's why our emails are typically available, and ask us questions.
Absolutely, and I thank you again, Desmond, for your time and for your important research. I'd also like to thank LabRoots today and our sponsor, Milton Biotech, for underwriting today's educational webcast. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care everyone and stay safe. Bye-bye.